All right, recording started. I'm not sure if they say that to you all, that you should hear that. All right, we have a good number of people in the room here, and like today is gonna be like, a kind of spatial session, where I'm gonna talk through, like the DNA cassette design. So the reason that we are trying to do this is because we are trying to move forward with the DNA synthesis and with the order through Twist, where they're giving us a pretty generous support. And if we don't do the design in time, we may not have time for them to do the synthesis and get back to us so that you guys at MIT here can work on your projects with the given DNA. So we want to make sure that you get prepared and order the right DNA. We will have some forms that you can put the order on for the MIT people. For the other notes online, there'll be a separate spreadsheet that you'll be submitting your DNA design to. So keep an eye on that. So with that, I'm going to lead this presentation. So it's going to be a pretty brief and quick presentation so that we have more time for us to discuss more. So let's get started.
Further, does anyone have any questions so far from this? Any concerns about this week? Because again, logistics are like, we don't have lab this week. The lab parts are replaced with optional self-pay experiments. So you get to go just like start some of the self-reactions we couldn't do last week, but really the time is for developing and working on this. So any questions in regards to that? Yeah.
It's not about the lab, it's about our orders. How many items can we order?
So that's a great question. So you can basically order as much as you want for the MIT and Harvard students, which is great. But the main part is that we need to know that you want solid like workflow and design. So which is why this week is very, very important. Again, Ice's presentation is going to be extremely important that if you have a good DNA design construct, if you have a good experimental plan, we can be able to help support that.
Yes. Yeah. So the number of fragments or the number of pieces doesn't tell you how much it's gonna cost. Is the length of the base. So if you're trying to order a big DNA, it could cost more than just one simple DNA. So let's move forward with this, and we're gonna figure it out at the end. All right, now let's get started. So with that, I'm gonna make sure that we cover all these three things. We make sure we navigate through the coding sequence. So we're gonna call it CDS. And we're gonna make sure that you know how do we do some kind of like reverse translation, turning the protein sequence into the DNA or RNA, and also do the codon optimization. We're gonna do it through Twist. There are several options there. Let's go with Twist because it's the platform they're gonna be using for the DNA synthesis. We'll make sure that we know how we put the coding sequence into the proper expression cassette. Just putting your DNA onto the plasmid or whatever is not going to work right away. You need all the complete parts in order for this gene expression to work. We're going to revisit like this kind of like construct. We need the promoter, we need the RBS, we need the CDS, and the terminator for any prokaryotic expression to work. And we would like, if you have time, we touch on the host DNA compatibility a little bit, very briefly. But maybe if you don't have time, we just skip over that, and we can assume that everyone's going to work on E. coli. All right, so a revisit on this is that we're going to work mainly on the plasmid DNA. So most of the information in biology or biotechnology is mostly stored in the form of DNA. So this DNA return to RNA and then protein. What we're trying to do is that we're going to take those DNA and put into the plasmid DNA, which is a lot easier to modify. If you're trying to modify the genome, it's going to be a lot harder. So for the time frame that we have, let's focus on the plasmid DNA. And let's frame this into a design, build, test, learn kind of concept. So with this, the things that you've been doing so far is that like you've been exploring the possible ideas through the literature review, right? You're going to be, and through that kind of like reading that you've been doing, we should make sure that you identify the actionable items. What does it mean by actionable items? It means like something you can do within the time frame that you have and with the materials that you're provided. So please make sure that make it like, some kind of proof of concept experiment. Think about it as like you're going to work on this project and you want to make sure that this is a project that you can do in the long run. Like let's frame it that way. And that will give you a much better kind of like starting point to design these things. So the thing that we have to make sure that we cover is the symbiotic focus. We want to make sure to identify the proteins or the genes that you want to use. You need to find the sequence and then design them and build them. So that's the first steps. We're going to focus on the design today, and you're going to be building them in a week or two. And then you want to put them into the action plan as well. Like, it's not just making a DNA and that's the end, right? You have to test them, whether it work depending on, what you have designed or not. And after you test them, you are going to take the data and like try to learn something from it. So keep in mind that the BTL cycle in the SynBio can be multiple rounds, but for the time that we probably will be doing just one round. So if you're ordering any DNA, keep in mind that like you may have time for yourself and for the company to synthesize that for you, just only one round.
Yeah, and by one round, it's like what the sense working out to is like 1 week of like very heavy on like full lab work. So for example, just in Anderson assembly lab, you might be doing something similar to that kind of scope project over the course of 1 week. So try to order as much as you can, plan ahead of time for this, but like just know that that timeline is gonna be 1 week for this kind of proof of concept. Generally how this goes about.
All right. So let's take some examples. So I'm going to show you 2 examples today on 2 distinct projects. First is a single chain insulin. Let's say I want to work on a single chain insulin randomly. It could be one of my projects. Let's take like a small step here to see what I can do by just working with these kind of projects. The second one is the neoleukin-2, which is something that is a designed protein, a designed peptide from David Baker's lab. This is published works, and what I'm trying to show you is that like how do we find them using like whatever tools you have, Google, PDB, UniProt, whatever. Like this is a mock-up framing question that I'm gonna like walk you through. Like can we simplify the production of these single chain insulin? Can we do it in the cell-free synthesis? In that case, like we need to think more about like some other context, and maybe if we wanted to, we want to put the GFP tag onto it. And that should be like help us for the measurement. So in a similar way, can we design a stronger binding? Can we use the current binding affinity of these neoleukin that we're still work on other things and try to tag them with the fluorescent proteins? No. For this kind of concept, I'm trying to bring this up just to show you that like, if you're trying to tag with multiple fluorescent proteins, then that means you have to take more time to design these things, right? You need to identify like which fluorescent protein you're going to work on. So my quick response would be like, if I have a very short amount of time, I'm not going to work with multiple fluorescent protein. Let's do like, let's make a lot of neoleukin-2 in state and try to screen with different inducible system, like in a way that like you're trying to make a lot of them. Like, in the bioproduction, probably like, you may have catch on that a little bit during the lecture that like sometimes if we're trying to make a lot of proteins, it makes a lot more sense to do inducible way, because you don't want to give too much burden to the cells during that growth stage. And then again, I'm trying to make it simple by just like put them into the GFP tagging style. All right, so these are the 2 concepts that I'm trying to introduce. Like one is like relevant to the cell-free part that we have been walking through. The other one is the bioproduction part, and you see one of these figure in your lab next week on the bioproduction side, in which like you're going to taste in different conditions, and that's what we can do in the genetic level as well. With that, one thing that I'm gonna revisit on is the cellular biology open language. So why do I mention these? It's just to provide you the link of like these kind of symbols that you may see from time to time in the course. The only thing that I want you to take away from this slide is this part, where like any genetic construct that you're going to work on in microbes, you need these 4 elements, which is the promoter, the ribosome binding sites, the coding sequence, and the terminator. So you can take one of those 4 and try to switch them around. This is like one of the good examples that they try to make different toggle switch using the SBOL. So technically you just like take different parts and combine them together to find a better toggle switch kind of. So you can use this symbol to represent whatever parts you're trying to work on. So if we were to work on these like single chain insulin, what should we do? The thing that you have to do first is you have to find the protein sequence of this single chain insulin if you were to work on it. It's not trivial, we're gonna do it together. 2, you have to design the DNA sequence. When you have the protein, you can't order a DNA synthesis for it right away, right? You have to turn it into, like do the reverse translation into the DNA that can express this thing. 3, you have to put into the cell-free compatible plasmid. You have to know what kind of experiment you want to do at the end so that you can put into the proper context that you're trying to work on. So in this case, you're working on a cell-free plasmid. You have to make sure that that plasmid that you're working on is compatible with the cell-free. And you're going to put them into the proper context you're working on. lastly, we want to make sure that you have the GFP fusion. If you plan to work on the GFP fusion strategy to measure the fluorescent level and then get that kind of amount of protein that you're expressing. So the reason I'm trying to stick to these fluorescent proteins because it's a lot faster to do the test if you do just the fluorescent readout.
One thing you could also do for your designs too, like just not to interrupt, is like you can put a GFP at the end after your gene to see if the whole thing has like folded properly too, which is a very common method that's used in a lot of protein design and protein folding to just ensure that it works.
Yeah. So the final thing for this kind of whole project is that we're going to put them into the cell-free expression system. So maybe like if we have time, we're going to try this week. But that's the thing that we do after we design and build, right? That's the test part. So with that, let's do a demo. Let's see.
Just make sure to share with your desktop now.
Yes. Maybe one. Let's go to the other slide, and I'm going to show the demo at the same time.
Yeah.
So this is another scenario that I'm going to walk you through. So if you were to work on the neoleukin-2, you have to do the same thing. You have to find the protein sequence, do the reverse translation. Again, this is why we want to work through with you, that it's not just one project that you have to do this. In any project, you have to find this kind of protein sequence and take them into the DNA sequence and order them. And then for this case, since we want to do the inducible expression strategy, you have to find some inducible expression plasmid and then ensure that you can design them in a way that you can put the GFP fusion to it so that you make sure you know how much protein you're making.
Does anyone know of an example of a common example of molecule used to induce the system? Like, yeah.
Lactose.
IPTG, lactose. Good.
Cool. Yeah. So, there are a lot of collections and we can go into, details on like how to find that. And again, like after this, we're going to check the option of how do we find the protein synthesis. So with that, I'm going to skip to the other screen. Let's see. Let me get out of that one. And
Any questions so far? This, do these workflows seem okay for everyone? Yep.
How would you recommend, we decide between which one to use, since we have a
I'll
short,
share the screen again.
since we have a shorter time frame.
Yeah. I guess it depends, like, for example, are you talking about which system is like cell-free or using like Ebola? So you can do either one, like either one's totally fine. Like generally the way we've done it most of the times is through the coli system, just because like we haven't really tried to hold on to cell-free. But you can also plan for Ebola too. Like again, you can express this plasmid in a coli-based system as well. So it should work theoretically.
All right.
Well,
Sorry.
just one quick thing, if you're gonna design it for a cell-free system, though, there are specific criteria you need to meet for that specific promoter and so forth, so.
All right, so I'm sorry for the small screen I have here. And this is I'm gonna start by going to the Twist website because like something that I want to show you later on is that you can create your own account, you can log into Twist and see what kind of options you can do. So in this case, you see that there are like the gene synthesis and you can do it in two ways. I'm gonna show the slide later, but what I really want to show here is that there are the part called My Vectors right here, where you can. Oops, sorry, I think I may have. Did I share the screen properly?
You're good.
Okay. Just gonna make
I know,
sure.
right?
Yeah, all right, can I take this away? Go to more.
And then hide floating meeting controls, fourth one.
Floating, there you go, all right. I'm not sure if I can find that again, but we'll see. All right, so in this panel, what you can go to is my vectors. So the options Twist provided you is that you can send your vectors and ask them to synthesize something, put into your own vector, so that you can skip the cloning step. So this is very convenient, especially for you guys who may not have a lot of like, cloning time. I would say time, because like they can do much faster than you. So, if you're new to this realm and you're gonna take more time than them, it might be better if you design it in a way that they can do it for you. So they have like a few options here. For the sake of time, it's sort of like I know some plasmid here. You can see my search function right here is still showing PET28. I don't know why they don't show that to me, but I'm gonna keep scrolling down. And I believe that would be PET28 right here. All right. So PET28 is one of the transcription or translation vector that can be used. Like a lot of people who try to make proteins use this vector all the time. The reason I come here is because I want some starting point. If we were to work on this DNA, ideal template. When you design a fragment to put into the plasmid, it's better that you know what plasmid you're working on. So I'm gonna go to benchling now and try to work on the DNA synthesis. I'm gonna make a dedicated folder. Week 10, 2025. All right. So again, this is what you will likely have to do. You're gonna go through choose file, go to whatever you've downloaded, take the plasmid, upload it here. You see that the plasmid look like this. This look very crazy because you're showing all enzymes and all enzymes. So I'm gonna make it smaller a bit, otherwise my computer is gonna go crazy. All right, I'm gonna leave it right like that. And we're gonna come back to the plasmid a little bit later, because what we want to do is to make sure that you find all the right proteins and DNA sequence you're designing for, right? So this is what the plasmid look like. It is a T7 system. It has T7 promoter. It has like operator, and it has something that is useful for the tagging. You can take a look further into that. And again, it has promoter. It has efficient ribosome binding site. So if you're using this plasmid, you don't have to find any RPS. And it has a terminator as well. So that means you don't have to find a terminator. If you're using the T7 system, go ahead and just use this one. All right, let's look into the single chain insulin. If I go and search for single chain insulin, I think I recognize this paper. So I'm going to go into this paper and see, can I find the actual sequence from them? So this is like a real kind of digging, like it's not easy. But when I look into this, let's assume that I read the paper already so that we go faster. When I go through it, I sort of see that like they have some PDB, like because they have the protein structure, they do the X-ray, so they must have the PDB information that was associated with this thing. So by having this, I'm gonna try to search for the PDB file. And this is like based on my, previous training, that I know that if they have the PDB file, they must have the protein sequence, right? Because there's no way you go to, make this kind of protein sequence without making this like protein structure without protein sequence. So I think I see something right here. W5WBM. This is the code that they gave me. They even hyperlink it. So now I'm here.
How convenient.
So right here, they said that it's a single chain insulin, and they have 57 sequence length. So with that, I'm gonna try to. I have to look for like where the actual sequence is, right? And if you go to the PDB, it could be different in a different format, but like this is the PDB. You go to sequence right here. You can try to download the files right here. If we try to download the FASTA file or FASTA files, whatever you call, it's going to give you this kind of information. If I open the file, it goes to SnapGene, but maybe we don't really need. There's SnapGene. Let's just display. So this is how it looks. It's very short. It's just this like short protein. And what we need to do is to turn this short protein or short peptide into the actual DNA sequence. So I'm going to go to twist. Oops. No, sorry, Microsoft, I need people. So I'm going to go to twist codon optimization. Thank you for sponsoring us. All right. With this, if you go to this page, it's a beta version, but it's pretty easy to use in my opinion. You can upload the file, you can upload the FASTA file, or you can start manually. And if you do manually, what they're gonna ask you is they want the name, which I'm gonna give them SCI. And the origin is gonna be whatever I just copied. And with that, click next, then they're gonna ask you a few things. Because you're trying to do codon optimization, you need to tell them what kind of optimization you want it for, right? So briefly, you can just click here, select all. You can select from the organism list. This is something that you need to do. No matter what kind of optimization you're trying to do, you need to give them what organism you're trying to go for. So Escherichia coli, apply. Let's think about, like, if I want to clone them into something special, I want to avoid some enzymes. I'm gonna put like some random enzyme here, equal R1, equal R5. It's gonna be pretty easy for them because it's a short protein. It's not gonna have too many restriction sites, but I want to make sure anyway that we sort of skip these kind of like restriction sites, because it might be something that we want to use as a strategy. BSA1 is one of the good example because it's the one for the Golden Gate cloning.
And you're doing E. coli for this one because your cell-free lysate comes from E. coli?
Exactly.
Mhm.
Good question. Yes.
Yeah.
If you're doing the human cell-free lysates, you may have to optimize them for the humans, and you have to use the human promoter, the human terminator, and the human rhizomatic sites.
Design considerations that you should put on your Notion page or whatever doc you're doing as you're writing this, like, okay, what system am I doing? Like, what chassis am I using? Like, these are all very important.
Yes. So I hit optimize, and I need to give them an email because they want to know who's using their tools.
Keep track
And-
of all of this, as well, so any TA that's advising your project can ask you, okay, what did you use to optimize your system? Like, what did you select? Where did you find this sequence?
I tried this a little bit before coming here, so I think it's gonna take like maybe 1 minute or two to get through this. So we'll just wait for them.
Any other
And-
questions? Ask away, because like, this is all like very valuable time to like.
If you're trying to use like. through more like human basis, how would you know which to avoid, like it wouldn't be eco or PV? Like how would you know?
So these these enzymes are based on something that you're planning to clone. Not exactly on like what you're trying to avoid at the end, right? So when you're trying to clone these things, if you use restriction enzymes and you're trying to cut something at the end of your DNA fragment, you don't want to cut something in the middle as well. So this is practically for the gene, the design when you do the cloning. So think about something that was like 1,000 amino acids. It's pretty easy for them to have some random sequence that can be recognized by your enzymes. So when you do codon optimization for the synthesis, it's good for the fragment to not have that part, right? So that you can do the design properly.
And you can also use enzymes that we already have too, right? Like, for example, we have ECR1 and ECR5, so it's like very easy to be like, oh, we have them, we can just go and cut them if we just avoid those sites.
So I skip a little bit, so it will return you this screen. And I just like download the file, and I will have, I'm not sure if the Zoom people will see my my PowerPoint here. Sorry, not PowerPoint, Excel. Can the Zoom people see my Excel file?
We can see the twist thing, not the Excel.
All right. Let me do resharing real quick. Just want to make sure that you see what the outcome looks like here. So the outcome of your file will have this thing, is going to be the sequence name that you put in and the final sequence that you've got from the codon optimization. So, and with this, you can copy it and just hitting Control-C. And I'm going to go back to the other screen again.
As he's doing Control-C, make sure you Control-C everything, or like just make sure you have your entire sequence. I've seen cases where people forgot the last two if they go manually drag it, so please double check.
So I'm coming back here to the PET28 sequence. Hopefully you can see my benching right now. I'm gonna go to the sequence right here and see what kind of design they have. So in this case, in this format of the PET28, it has this HTG right here, which is something I'm familiar with, which is our start codon. If I'm trying to put anything into here, it should go after the start codon. One thing I can show you real quick about this vector is that they have a lot of the tag system right here that you can follow through. So it has like the left hand is tag, the right hand is tag. So you can cut at some spot right here just to make sure that you have like the right thing. So I'm gonna assume that I'm taking only the ATG and, you know, ignore everything. Maybe I'll keep this.
Does
Yeah,
anyone know
his
why
tag.
his tag is useful? Okay, his tag can be used for purification, so like you can go in and decide to get a certain column that like actually essentially goes and purifies based on the his tag. I forgot the exact specifics, but it can be very handy for protein purification.
So before replacing this, I want to show you one thing real quick, that the translation cassette goes from here to here. I don't know why they give me that random thing.
Have I changed something this week?
So this is a coding sequence. So right now you would take this vector and just like go and do whatever, you know, expression, you're gonna have this kind of protein, which may be something we don't really care. But if you replace this thing with your protein sequence that you just got from the codon optimization. Now you have a new coding sequence. Again, when you paste something, copy paste, I try to make sure that I did the pasting properly. So if I search again, I did Control F and search for it. It's actually showing up right here. So this is the SCI, the single chain insulin that I just designed and pop it there. And it is very important that it is in this T7 promoter. It has this RPS and it has some T7 terminator right here. Once you put this plasmid into a cell-free system that has the T7 promoter, it's gonna express. Now, we're missing one thing in our plan, which is the GFP fusion, right? We didn't do the GFP fusion of this yet. So I'm gonna go back to our previous information on the GFP. I believe I used to have some SFGFP in here. Let's see, SFGFP uprising. And this is the GFP sequence that I used to have. So I'm gonna copy that and make sure that they're still in frame. So one of the keywords that is important for the designing of the G cassette is that they are in frame. What does it mean? When you read, when the ribosome read your sequence, it should read in a 3 base codon. But so if it's out of frame, it's not going to be the same piece of protein. So now that I'm pasting this SFGP on top of this, like, you know, single chain insulin, it's going to be a fusion protein only if it is in frame, right? And one thing in extra that I'm going to do is to get rid of this start codon. Why do you want to do that? Because if it has a start codon, there's a chance it's going to express and make just a GFP, right? So if this part upstream here turns out to be something similar to ribosome binding site, then you have the GFP expression, just the GFP, not the fused SCI GFP. So that's why, like, when people do protein fusion, they usually get rid of the methionine, the start codon, just to make sure that you make the chance of like translation of that particular protein into like near zero. And here, I don't need to stop codon. I'm gonna delete one of them. It's not too bad if you have 2 stop codons, because it's gonna stop anyway. But with that, now I have my plasmid. How do you design this thing? Not quite clear, right? Because if you design them in this kind of piece, you may have to do some PCR to put them into this overlapping site. Think about the Gibson assembly. The plasmid that you have are anything but the sequence you just added here, right? I'm gonna do some inverse selection. So this is a piece of DNA that you have before as a PET28 plasmid. The thing that is not highlighted here is the thing that you're trying to put them in. So when you design the DNA, it might be good for you to include something upstream and downstream if you're trying to do the Gibson assembly, because you need that kind of overhang sequence so that they match up when you do the Gibson assembly. If you want to do Golden Gate, if you want to do restriction ligation, you may want to add some enzyme cut sites into those things. So since we're doing the synthesis anyway and adding 5 or 10 bases, or let's say 20, doesn't cost that much compared to the thing you're trying to synthesize. So make sure if you want to do anything downstream, and it's better that you don't have to do another PCR round, try to take advantage of these synthesis so that you can go faster. All right, I think for the interest of time, I'm gonna show only for this one. But I'm gonna show you how I find the interleukin-2, just for the sake of, sorry, neoleukin-2. Just for the sake of like finding things. So this is the paper that I show you on like where I found the neoleukin-2. And technically, this paper is about the split neoleukin-2, not the original neoleukin. So if you want to express the whole enzyme, you may have to like go find the original one. Around here, what you can look for is that like, look for the references, because if you are looking into the protein and it's not the protein that was developed in that paper, it's probably, appended onto one of the reference. So, let me do some search here. Let's see. I didn't expect this PDF. I want it to be easier to search.
Oh my God.
That's,
That's that host. 32 is still high, it's halfway there.
all right, I give up. Let's come back to Google. Neoleukin.
Yeah, he's doing it live to show you, like, this is checking the
15.
process of
This
like,
is the
yeah.
process. 2019, that's when the paper came out. The novel design of potent selective limit of IL-2. That's where it's come from. And I want to see it on the publisher site, so it's going to bring me to Nature. Oops, a lot of cookies. Then this is, the sequence of what they were trying to design. Usually, if you're looking for the actual protein sequence, is a lot of information, so they usually don't put it into the paper. They put it into the supplement. So you can look for the protein sequence that should be in one of the table right here. You see maybe like 30 supplementary figures. All right.
Yeah, makes sense.
And if you keep scrolling down to that final screening, you see this neoleukin-2/15, which is like, chimeric motion. They give us, some kind of like shortcut by bolding them. So that's the actual thing that they taste. Take this, copy the sequence, and do the same thing. Put the protein sequence into the codon optimization tools, get the DNA sequence and put it back. So like this PET28 is like one of the things that could be used as well, because it's based on T7 and it has this like operator where you can use IPTG inducible system on it. If you want to change it to something else, I recommend going to, iGEM. It has a lot of parts. It has these like catalog of promoters and ribosome binding site. This is like, some RBS part from Madras collection. There are some, other RBS from this collection here. They will have some relevant information like whether it's high or low. There are also these part of, the inducible promoter. There are more than one. You can look into that. If you think of like designing different RBS or promoter, you can include that into your DNA synthesis as well. So instead of like ordering this piece of coding sequence, you can include up to the upstream site and design your cloning differently. Or you can even tell Twist if you're using something that they have to cut right there. And please put the design sequence into that point, and they can do that for you. So that's all I want to walk through. I'll go back to the slide and like, summarize what we've been talking through a little bit. And let's see. One sec for the Zoom people. Changing the screen share right now. All right, so coming back to what we just talked, we want to make sure that we check the option for the DNA synthesis. And the options that we have are gene fragments and clonal genes. So clonal genes means you can order them and clone into the plasmid already. And what you're gonna get is a plasmid that you can put into the bacteria of interest or like organism of interest. This is an option that you have. Depending on like what kind of option you're gonna go through, they may have different workflow. So if you order the clonal genes, of course, it's gonna take longer because you have to synthesize into the gene fragment and put them into the plasmid. Technically, it could be as small as just 2 business days. And in the options, there are a lot of vectors. I just, scroll through a little bit of the vector options. They have like more than 20 that you can choose from. And, you're just gonna put those fragments into the plasmid. If you want to order in the gene fragment strategy, you have one important question right there, whether you need an adapter or not. So why adapter? It's because like it's easier for them to do the synthesis with the presence of adapter. There are like a lot of info included, but like that will make your DNA having some small chunk of DNA at both ends of them. And sometimes it's important to make sure that you don't have that thing at the end. If you're trying to do Gibson, then it's not the overlapping sequence you're trying to use. So you have to omit that no matter what. And lastly, you have to make sure that the sequence were 300 to 5 kb, because something smaller or larger than that, Twist doesn't provide a good like workflow for making that. So if you want something more than 5 kb, it might be better to design 2 inserts that overlap for the Gibson instead. So that's all. Summary is make sure that you go through the design and build and be aware of the taste and something you want to learn at the end. And that's it. Awesome. Let's go to the other presentation if we have time.
Yep. That's right. Do you want to potentially either screen share or like join the Zoom and then put it on here? And then if I just want to keep that up, we can keep it there. Awesome.
Yeah, if you.
Yeah.
That would work the best.
Yeah.
Oh, okay.
All right. So I'll start going through the questions in the chat briefly, just to be aware of the time. All right.
Okay.
Ask a question about recording is recorded and we'll share that as soon as possible.
Awesome. And then Ice, can you switch to that screen?
Show you that. Oh. All right. So. Come on. Right, we're here.
Awesome. Great. Can people that are currently in the Zoom hear me as well?
I think they can.
Cool, I see some noddings. So awesome. Hi, if you don't know me, I'm Ren. And I'm gonna be helping run the biomaterials lab that's gonna be happening at the very beginning of May. And so I just want, in this lab, you guys are gonna get to grow some fungal objects and take them home. So I want to give you all the opportunity to design the shapes that you're interested in growing. And so I just wanted to talk through that process a little bit, and then hopefully get some of the designs from you in time that we can fabricate them. So if you've never seen fungal objects before, this is kind of what they look like. And so the whole process takes about 2 to 3 weeks to form. And so we're going to be coming in, you guys get to design the shapes that you're interested in growing. And then during the lab, I'll have some setup material. You'll get to like pack your molds, and then we'll let them grow, and you guys will get them kind of around finals week, ideally. And so just a brief overview of how this works. This is with EcoVato's Grow It Yourself kits, where this will be on the back end. We'll basically prepare the substrate, which has this fungus called Ganoderma in it. We'll let it grow for a little bit, break it up, and then you guys will come in on this step 2. You'll get to fill your growth forms, and then we'll let those sit for a while. We'll take care of that, and you should walk away with some cool fungal objects. So if you want a custom object, You have a few options, so I'll talk you through one of the options that you have, which is this 3D printing into silicone mold process. So I have some of these here that I'm gonna pass around to you guys, but basically, here you can take a look. If you have a design that you're interested in, say like this component right here, and you're proficient at 3D design and whatever software you're interested in. Your goal is to make a positive mold that is that shape, but it has a lip so that we can fill this mold with liquid silicone to create the negative version of that shape, right? Does that make sense? I see some nods. Okay, cool. So you would basically make that shape with a lip so that we can fill it with silicone, and that gives you a negative version of the mold, which this is what you would pack with the fungal material. Any questions about that? Cool. You can also, if you're interested in just experimenting with different like molding techniques, you're more than welcome to show up with a mold of your own design. Some people make them out of cardboard. If you're really fancy and you have access to one of those like vacuum forming plastic machines, you could do that if you wanted to. If you don't want to go, if you're not interested in kind of creating your own design, I have some existing molds that I will be bringing, so you could pick from those. But if you are interested in making your own shape, then this is what I need from you. I would say that you can get down to, I tried to provide some of the constraints, which is kind of something within either like 2 by 2 by 2 all the way up to 8 by 8 by 8, which I say because that's the max of a 3D printer. For your design, unless you want to fabricate it differently. But I would say that something kind of in between those two is gonna work out the best in terms of your dimension, kind of like a normal distribution. Any questions?
Please don't make an 8 by 8 by box. Out of 30 box. Please don't do that.
That's true, yeah. So if you guys can submit, if you want to go this route of 3D printing, I need your designs sent to me. I'll look them over and just be like, yeah, this makes sense, this looks good, this will work, or I'll send it back to you with some notes for edits by April 20th. Yes.
Can we send our designs over Slack or?
Yeah, that's totally fine. Yeah, you can also send, just get it to me somehow, whether that's over Slack, whether that's over email. And then I will check it and we'll get it made and it will be there ready for you. Come the biomaterials lab.
Oh.
No questions? Yes.
Can we see the final result of like the biomaterials, like how it looks?
Yeah, it kind of looks like, I also have some examples of this in the upstairs lab that I can do, if you want to see them, we can go up there right after this.
Are you gonna grab them?
Oh, yeah, if you wanna grab some and run back down.
Yeah, we were just running demo day for the media labs. We have a bunch of those samples as well as some of the open trends are, so.
Yeah, they're like, they're fairly sturdy, a little bit, like some of them can be a little flaky. It'll depend on in my eyes, I think the surface area to volume ratio gets kind of critical for these things, so. I'll take a look at your designs, but be aware that these are made with wood chips. And so if you think about like this mold, for example, which you might struggle to see in the back, but it has little tiny circles. This was for biocement with bacteria. And you wouldn't be able to achieve this level of resolution with the mycelium material just solely because like the wood chips can't fit into these little holes. They can't. So you're looking at stuff that is more on this scale. I see a question in the chat. Besides rapid growth, what are the mechanical properties of this biomaterial? If you look up Ecovative, grow it yourself, they'll have a table on their website that gives you some of the like technical measurements about the mechanical properties of this material. I don't remember them off the top of my head, but like Ronan's gonna bring down some samples. They feel something along the lines of like cork. if you've ever felt that, or like a really spongy particle board, and then after you bake them, they kind of become like styrofoam-ish, styrofoam adjacent. So they're really great. I would recommend something along the lines of like a planter or a pencil holder. Yes.
So
You in
my
the,
final project revolves around like actual like fungus, actually. I was wondering if like maybe I could help incorporate this into my final project as well, or is this just like a separate lab thing?
you're totally welcome to incorporate it into your final project.
Okay.
Right.
Yeah.
Why not?
Yeah, it's quite huge. You can do whatever you want me to say for that, so I just come up with a good experimental plan.
This is, I mean, the title of the course is just how to grow almost anything, and this is a very relevant part of growing things.
Please don't make it 3 ion.
Is there a minimum recommended wall thickness or feature size?
Yeah, so I would say if, I'll check over people's designs, but if you're doing anything smaller than like half an inch, you might run into some stability issues. Or feature size, I would say experiment with it. I can't give you an exact answer, but the more resolute, the more challenging. Oh, thank you for putting them in the box here. Now I'll pass this around and you can, that's just a straight cylinder, so you can feel the material, and then this is I'll show it on the camera, if you have your camera on.
Is this
Oh.
the owl?
Cool, hello owl, this is like a little planter pot.
That's forgetting about this. I'm like so much.
Yeah, but then here, I'll also pass around some of the smaller ones, and you can feel the difference between the like smaller entity and the bigger cylinder.
Don't eat it.
You don't eat it. I will say don't eat it, but it is technically, this is the reishi mushroom, which is a very common edible mushroom.
Not cooked
And
yet.
if you don't, so these are all deactivated, they were baked at 200 degrees for a couple hours.
Oh, so it's.
But if you don't have to deactivate them, and if you don't deactivate them, they will actually grow mushrooms.
Whoa.
So yeah, any other questions?
What's the purpose of using the silicone? Why don't we just 3D print the?
Great question. So you could 3D print it, but trying to get something, it'll like depending on the shape. This is a very solid thing. The silicone, what's an advantage of it is if when it's time to demold it, I can just do this. So it's flexible and I can easily get it out. You could theoretically put it in this, and that one, for example, some of the circular ones I made in beakers, but I did have a challenge getting them out of a solid mold.
The other way you could do it with like more materials, like you make a 2-part mold, and like you have, let's say, like a circle going in and a circle out as pegs, to, like keep them. But again, it gets trickier and trickier than just putting in silicone and then
Yeah.
doing.
You're totally free to experiment, and again, just send whatever you're thinking to me, and we'll look through it together, and I'll be like, this is feasible, this seems challenging, etc. But yeah, great question.
Awesome. Sounds good.
Great.
Right.
Thanks so much, guys. Okay,
Yeah.
this is hopefully.
They were chatting that the life lab at London will be trying this lab as well. So.
That'll be exciting. Yeah.
Sturdy and resistant materials. Yeah, there are other resistant materials. Biocement is a common one that's made with bacteria. I have no idea. Yeah, biocement people make stuff out of algae. There are people making things out of SCOBY, which is synthetic community of bacteria and yeast. Which is like the weird little material that grows on top of kombucha. You can dry it out. People use bacterial cellulose. You can get like leather-like properties. But I would say fungi are at the kind of cutting edge of the material side of stuff that you're gonna see. Are there any other questions in the chat?
Cooking process. If they're relevant, these are more questions.
Oh, man. Yeah, you can also, we did do some experiments about silicone printing, but But I don't think we have this, like, I don't have a silicone mold, so you would have to BYO silicone mold.
Like you can also use like TPU or something like some flexible filaments, but again, it's probably just easier using silicone.
Yeah, so if you have experience, and again, if you want to experiment with making your own mold, you're so welcome to. It's just that we have access to classical PLA 3D printing, so, and liquid silicone. Cool. If you have any other questions, just message me on Slack or send me an email.
Sounds good. One thing before we start the recording, I see some question about the start codon. This is very important and I want to address it to everyone, both in the global listeners and also people in the room as well. Some proteins you find out there may not have methionine, may not have the start codon included because they were processed. But if you were to express them in any system, they need the start codon. So keep in mind, like try to make sure that you have the start codon and the stop codon. That's another important feature of the coding sequence. You know, like, again, 4 things: promoter, RBS, coding sequence, terminator. You need all those.
Let me add something else to this. I want to see a diagram of people just drawing this out, and be like, this is where my terminator is, like where everything is in your map, like either an annotation on Benchling, or you just write down on this paper that you have these drawings. The symbols on one of the slides that was shown, use that like naming and labeling scheme for this. Like, please do it as like straightforward and like standard as possible, because it makes your job easier as communicating it. It makes our job easier to knowing how far you guys are and what you need help with, and just make sure you do your best to stick with some of this process. Any questions on that? Yeah, we want to make sure everyone's on a good stop for ordering DNA on Friday. I know it's an aggressive timeline, but we have roughly 6+ hours of like support for like office hours this week, and we can add more based on PAs as reach out. So. All right, cool. We'll be here for a little bit for office hours, but yeah.
Cool. Then I'll end the recording right now. Thank you, everyone.
Hi, Ice.